Hello my dear students, welcome to the video. Today we are going to learn structure of microsporangium in flowering plants. The flowering plants are also called as angiosperms. They reproduce sexually. The sexual reproduction occurs through the fertilization of the male gamete pollen and the female gamete ovum. Leads to the formation of a single cell called zygote. Zygote undergoes cell division and differentiation to produce a new plant. Flowers are the reproductive structures in angiosperms. They possess the reproductive organs. The male reproductive organ is called andrecium and the female reproductive organ is called gynecium. Andrecium is composed of a cluster of individual structures called stamen. A single stamen has two parts, a long, slender, filamentous region called filament, and on the distal end of the filament there is anther attached. The proximal end of the filament is attached with the thalamus of the flower. A stamen is having a structure called anther. This is the site of formation of pollen grain. Anthers are bilobed. That means it has two lobes. The two lobes are attached together through connective tissue in the center. Each lobe of an anther is having two theca. A longitudinal groove divides a lobe into two theca. So it is known as dithecous. In each theca there are one microsporangium. So altogether in an anther there are 4 microsporangium present. Microsporangium is the basic fundamental unit of the male reproductive structure in flowering plants. Since there are 4 microsporangium present in an anther, it is called as tetrasporangiate. The cross-sectional view reveals that an anther has 4 microsporangium located in the 4 corners of an anther, of a tetragonal structure of an anther, as we can see in the micrographic image given in the slide. The microsporangium are having a circular outline composed of multi-layered cell walls. In the center of the microsporangium, there are tightly packed cells called sporogenous tissue. Microsporangium is the site where the sporogenous tissue undergoes meiotic cell division followed by differentiation to produce mature pollen grains. Once the mature pollen grains are produced, the anther walls start to split up to break open the walls through the longitudinal groove to release the matured pollen grains for pollination, as it is given in the second image. This process is called anther dehiscence. The anatomical structure of a microsporangium shows that it is composed of 4 layered wall, and in the center of each microsporangium there are sporogenous tissue. The walls of a microsporangium is composed of 4 different cell layers. They are epidermis cell in the outer layer, endothecium, middle layers and tapetum cells. Epidermis cells are providing protective role to the microsporangium. They prevent the water loss from the microsporangium. They provide mechanical support to the anther walls. Endothecium is the second layer. It is also involved in the providing mechanical support to the anther wall. Along with the epidermis cells, endothecium involved in the process of anther dehiscence. Middle layers are short-lived cells. They occur in 1 to 3 layers in a microsporangium. At the time of microsporogenesis, the middle layers degenerate to disappear. By the end of the anther maturation, the middle layers get completely disappeared. Tapetum cells are the innermost layer of the anther wall, of the microsporangium wall. They are involved in the nourishment of the sporogenous tissue, as well as they secrete sporopollenin. Sporopollenin is a biopolymer involved in the formation of pollen wall, the outer wall of the pollen, called exine. Sporogenous tissue, initially diploid in nature, undergoes meiotic cell division during microsporogenesis to produce microspore and get developed into a mature pollen grain. And the anther dehiscence leads to the release of matured pollen grain to the exterior for pollination. This is all about the structure of microsporangium. We will summarize the important topics. A microsporangium is a basic fundamental unit of the anther where the male gamete pollen grain formation occurs through the process called microsporogenesis. At a time, there are 4 microsporangium conduct microsporogenesis independently. Millions of pollen grains are getting developed in a single microsporangium. It is present in the anther of a stamen. Anther is a bilobed structure, dithecous and tetrasporangiate. The microsporangium is made up of 4 layered cell wall and consists of sporogenous tissue in the center. The 4 wall layers are epidermis, endothecium, middle layers and tapetum. Epidermis involved in the protective function, endothecium provide mechanical support and involved in the anther dehiscence along with epidermis. Middle layers are short-lived. By the process of pollen maturation, middle layers get disappeared. Tapetum cells involved in the nourishment of the sporogenous tissue. The walls of the tapetum cells break open to release the cytoplasm. The cytoplasm flows towards the sporogenous tissue and this acts as the nourishment to the sporogenous tissue. Tapetum cells occur in multinucleated manner due to the entomitotic mechanism. This is all about the structure of microsporangium. I hope the concept is clear to everyone. Thank you for watching.